All right. So no introduction. We're diving right in. Okay.
Tout le monde te connaît. Tout le monde te connaît.
Yep. So yeah. Okay. So here I am. I'm back, and I'm after Michel telling you about imaging in thick tissue. So as I said, we'll dive right in because this is not a talk about my research, actually, because Michel gave so many good examples. I took out everything that was specific to my lab, and it's really a teaching presentation. So the point of many of the things we do in at Servo or in neuroscience in general is that we're trying to understand the interactions between various parts of the system. And it's true in many other areas of research like immunology. Well, we need to worry about the immunological response, the nervous system response, the vascular response, even in some cases, the bone marrow, what happens in the bone marrow. So it's not the components that are critical, it's really their interactions, and the interactions typically translate into the need for in vivo imaging, because it may be true that if you study cancer or other things, you can take biopsies, but in the nervous system, we don't really work with biopsies. First of all, if you were to do a biopsy, you would take tissue out that you would take it out of its functional area and put it on your desk. Well, that's dangerous because the brain has no cells to spare, really. Contrary to cancer. In cancer, you're trying to take stuff out, and the things you're taking out, you want out anyway, and then you can study these cells. But in our case, we're trying to understand the normal behavior of the brain, so we want to leave the cells there. And at the same time, even if we were to take them out, it would be meaningless because it's not part of the network in which it functions. So we really need to do live animal imaging very often, or more generally, thick tissue imaging. So imaging in tissue that is mostly intact. So let's look at this. So live animal imaging means thick tissue imaging. So what do I mean by thick tissue? What is tissue, really? So find the imposter on this slide. So what behaves like tissue here? There's a brain, skin, a petri dish with cells, and a glass of milk. So which one is the imposter?
The petri dish.
The brain.
The brain, the petri dish.
Petri.
Petri dish.
The what?
Petri dish. Petri.
It is the petri dish. Okay, the imposter is the petri dish. And why is it the petri dish? Because the petri dish does not have the key properties that are a problem for us in tissue. It does not scatter light, and that's what I'll talk about for several slides. So when we shine light on tissue, I think there's a, how do I do that? No, okay. Okay, so we can have specular, if we shine light on tissue, light can be reflected from the interface. It can enter the tissue and be absorbed. It can enter the tissue and then come back out. Actually, it can enter the tissue and be scattered, and then it can enter the tissue, be scattered, and come out of the tissue. So these are four different phenomena. In particular, if you look at this one here, specular reflection and diffuse reflection, just to make sure that you understand the differences, specular reflection occurs when the light hits the interface, so air tissue interface. The tissue has an index of refraction. I'll get back to that in a second. And the light is reflected really from the surface. So it does not enter the tissue. If you were to try to make an image with this, the only thing you'd get is information about the index of refraction at the interface. On the other hand, this diffuse reflectance, the light enters the tissue and then comes back out. So those photons do have information and are useful for imaging. This is essentially what Michelle talked about for some time during her presentation. Okay, so moving on. I will describe what the challenges are when we do tissue imaging. This is something that probably affects many of you or will affect many of you. So we'll discuss scattering, or how light scattering is preventing us from imaging, so or sometimes helping us. That may be a surprise at some point, but we'll discuss that. We'll discuss absorption, how we can access organs when we want to image them. Eventually, we want to see something, so contrast is always a big issue. Because we're doing live imaging, we will often want to do live imaging, then movement is sometimes an issue. Something that is often forgotten is that live tissue means big tissue. So we're trying to get not some anecdotal evidence at a given point, but really information about, let's say, the animal or the whole brain. So we can't just stick our microscope in an area and hope that this is going to be the right information. We have to sample the volume, and that's an important aspect. And finally, the analysis is also something that's that we have to worry about. So as I said, let's dive right in. So scattering is the presence of scattering in tissue is what prevents us from imaging thick in tissue. So what is scattering? Let's discuss scattering. So we know one thing about optics. We know that the index of refraction affects the propagation of light. So in this case, if you look at this picture here, you have water here, and you have a pen and here, because the pen is outside of the tissue at the top, and the tissue of the water, and in the water below, you can see that the light doesn't propagate the way, and the light doesn't propagate the same way, and therefore the pencil that we're trying to look at appears to be broken or distorted. So the index of refraction is really what is causing the problem, because light interacts with the dipoles, the electronic dipoles, and the dipoles are really responsible for the index of refraction. So how does that apply to tissue? Well, if you have just a thin slice of tissue, with a homogeneous slice of tissue, then the index of refraction is homogeneous. The light is refracted at the interface right here and here, and and here. And this refraction, we know, this is the one I just described in the slide before. But the cells actually have an index of refraction that's not the same as the surrounding environment. So really, the photons are traveling and are seeing several small interfaces everywhere. There's one here, there's one here, there's one here. And each time they can enter or be reflected, or both, really. And the overall light that we are seeing is the sum of all these rays, or all these photons that are now coming in all directions. This is a problem that it doesn't come from the same direction, because in practice, I would assume that if the light is coming from, if I measure all the light and I can determine where it came from, we call that imaging. I would say, okay, if there's all this light coming from this point, then at this point there was this cell. But now it's difficult, because it appears that if you look at all these rays, then it appears that the cells are really anywhere in here, which is true, but it prevents us from doing high resolution imaging because the scattering is affecting our ability to create an image. So the way we characterize the scattering in tissue is with the scattering coefficient. So it's called μs, and it represents the number of collisions per second. So typically in tissue, the scattering coefficient is, I would say, 100 collisions per centimeter, or approximately 100 microns between collisions, unless I'm mistaken. There's 10, 100. Yes, that's it. So the scattering coefficient tends to be larger at shorter wavelengths and smaller at higher wavelengths, simply because if the wavelength is short, then it will be sensitive to things that are finer, whereas if you have a large wavelength or a long wavelength, it won't see the small perturbations as much. So it's a mild decrease with wavelength, but still it's a good rule of thumb that longer wavelength usually mean smaller scattering coefficient. This reference right here is an excellent reference. I'll give you all my slides at the end, but this is a very good reference with tons of pages and pages of tables for typical scattering coefficient. So if you need that, you can look it up. Okay, so what this means for us is that the light, as the light propagates, we will consider that it's bouncing off cells and scattered elsewhere. And so when we shine light to a given area, it will exponentially remove light from my illumination. So if I'm illuminating at the very top surface, then I'll be fine. But if I'm imaging 100 microns below, I will have lost approximately more than half I think I'm going to be down to 36. So that's 64% of the light will have been scattered away by the cells. Okay, so illuminating, it's a problem because my light doesn't make it to my focal spot. Detecting is also a problem because when the light is emitted, I would like it to come back to my objective, but on the way back, it may get scattered outside of the detection cone of my microscope, so of my objective. So that's no good. How do we get an idea for these numbers? We do have models, engineers and physicists have models for propagating light in tissue. There are a lot of great resources. The best one is called MCML from Li Hong Wang. But my group has made a small Monte Carlo based calculation in Python that is also available. And I think it's actually simple to use. So if you need numbers, you can take a look at this or talk to us. We'll give you more information. So how do we minimize the effect of scattering? We can use longer wavelengths. Okay, so you'll see, I forgot to say that, but you'll see at the top of my slides, I have a description of the problem. Here I'll have what is the challenge, description of the problem, and then solutions. So you can follow at the top if you get lost. So now we're in solutions. So if we use a longer wavelength, the scattering coefficient is going to be smaller. That's good, but it's a mild effect. We can use multi-photon excitation instead of single photon. I'll get back to that. I was supposed to have done that on Monday, but I have a few slides on that later, but I will discuss that. Okay, so we will see that two-photon microscopy has a significant advantage over one photon excitation for thick tissue. Okay, keep that in mind. I'll get back to you in a few slides. Okay, so in addition to moving the photons away, they will also make the scattering will make the focus not as good. Okay, so if we think of the wave entering the objective and being focused, it should be a nice wave or a spherical wave, a wave that focuses to a point, but that's not quite what happens. Because of the cells, we talk about scattering, but we can also talk in terms of distortion of the wavefront. These are two mostly identical or equivalent descriptions. But the point is that if we discuss in terms of how the cells are modifying the focal spot or the wavefront, it is possible to correct for this aberration. This is called adaptive optics. So what we do when we do that is that we know that the cells, the cells everywhere are modifying the focal spot. So they are distorting the wavefront like here. So if we put a mirror that imparts the opposite aberration, then as it propagates, the light will be focused better. This is an advanced technique, but this is a fancy technique that I wanted to discuss to say that it exists. So this is called what's it called? It's called adaptive optics or adaptive optics microscopy or wavefront compensation from adaptive optics. So this is something that people are looking at to try to compensate for scattering. And the other option, if you can't compensate for scattering, is to make sure that there is very little scattering. So there are various techniques that are used for clearing the tissue. And I could talk about that for hours if this was the subject of the talk, but it's not. So these techniques that are used can actually essentially remove or homogenize the index of refraction inside the tissue while keeping the structure intact. So they essentially remove the lipids from the tissue and replace that with a gel that has an index of refraction the same as the rest of the molecules. So in the end, you get something that is essentially transparent. And this is used. This has been used for the brain, for the spinal cord, and any other types of tissue. Okay, so in this, if you don't mind imaging, obviously the not the animal, but the specimen has to be dead because it won't survive this clearing procedure. But if that's okay with you, then that's fine. But because you do have the intact structure. Okay, so now absorption. Okay, we talked about scattering, but now absorption. The absorption in tissue for us is mostly by blood. Michelle showed how she's using that to get information, but for us, for me at least today, this is going to be a problem, not a solution. So as discussed before, again, there is a lot of absorption. There is a lot of absorption due to blood on the lower part of the spectrum. Notice how sharp the absorption is when we go above 600. So the absorbance is an exponential number. Notice that the absorption at just below 600 is in the range of 10 per centimeter. Whereas if we go to just above 600, the absorption is 100 times less. Okay, so absorption is a very significant problem in the low part of the visible of the low range of the visible wavelengths. Okay, and if we go too high, then water is starting to absorb. So there is a window, a good window between 600 and approximately 1200, a little bit higher, where absorption is minimum. How is that affecting us? It's affecting us because if we try to do live animal imaging or thick tissue imaging, there may be blood in the way. As we shine the light we will get some of the illumination to be absorbed, but also on the way back, when the light is trying to travel back, it may get absorbed by the vessels. It's mostly the vessels that are responsible. So I'm talking, but really, how bad is it? So if we look, this is me with a red laser pointer. And you can see that if I shine the laser pointer through my finger, you easily see the light through. I'm using a green laser pointer and shining through my finger. Well, I'm not shining through my finger at all. All of the light is really absorbed in the first few hundred microns of my finger, and the light does not make it through at all. So using red wavelengths is a very significant and an excellent method to avoid absorption in tissue. And it's not always a choice we have. You have heard about GFP and GCAMP and all these things. GFP is a great fluorescent protein that has a lot of applications and has been used in tons of transgenic animals, and it's all of many fluorophores or calcium-sensitive fluorophores are based on modified versions of GFP because it's well known. That doesn't make it a very good fluorophore. It just makes it a fluorophore. But in live tissue, it still works, of course, but it's not ideal. So red fluorescent proteins, you've heard of that from Robert Campbell a few days ago. But the point is that anything that's redder, it's mostly better. Okay. So second problem. So we talked about scattering. We talked about absorption. So contrast and labeling. Okay. So once we have figured out that there's stuff in the way to get the light to the part we want, or to get the light from the focal spot we want, we need to get some sort of contrast. Contrast in live tissue is not necessarily easy, because if you think that you can use, let's say, fluorescent dye, there are many dyes that are used in Petri dishes or in cells. You incubate the cells with the dye, and the dye will eventually penetrate the membrane or attach to the receptor at the surface, et cetera, and then you'll get a great labeling. Well, in tissue, it's not as simple as that. You need to get the dye to the cells, and as simple as this sounds, it is not that simple. Okay, so it's possible to use contrast mechanisms that are endogenous, so that only rely on things that are already in the cells or in tissue. And there are other ways to get fluorescent labels inside the cells. So fluorescent labels, if you can use fluorescent proteins, you will use the cellular machinery, and there are tons of strategies that you have seen in other talks this week, where fluorescent proteins can be injected with a viral vector that will infect the cells, and then the cells will start producing this fluorescent protein based on the promoter, etc. The point is, you can get these fluorescent proteins inside the cells with various strategies, and I'm not the expert for that. So I know that it's been discussed at great lengths from others this week. So also that's it. Okay, so we discussed, I said just before that we really want to have a as red as possible fluorescence, to make sure that we can get the light and collect the light from to and from the focal volume. So let's take a quick pause, because this is the right moment to discuss two-photon microscopy, which I did not discuss on Monday. So when we do one photon fluorescence excitation, what do? We come with a laser that has, let's say, a blue wavelength, and then it will take an electron from the ground state. It will excite it to the excited state. It will just decay a tiny little bit, and then it will go down to this ground state by emitting light. Okay. What this means is that if you have, I should have used a different color. Let's assume that this is green. Really, if you take blue colors to excite, you would get typically green fluorescence. But the beauty of the two photon excitation is that it is actually possible to take red photons that don't have quite the right energy, but you take two of them to excite the electron or the molecule. So if you can get enough photons per area near the focal volume, then sometimes the flux is going to be high enough, and two of them will work together to excite the electron to the excited state, which will then essentially decay to approximately the same state, but not quite, and then emit light at essentially the same wavelength that we would have used for one photon. But the beauty here is that we have used a color that's redder. Okay, so this effect, by the way, was discovered by Maria Goppert-Meyer, and the cross section, which I discussed on Monday. So the cross section is how big a molecule looks to a photon. So the cross section is typically on the order of 10 to the minus 6 per square centimeter. For two photon excitation, it's not measured in area because the cross section depends on the amount of photons. So it's the cross section in two photon is measured in GM, in Goppert-Meyer units, and it's typically 1 Goppert-Meyer or 1 GM. And if you take our goal when we do multi-photon excitation is to get the final cross section is really the intensity of our laser times this cross section, this 2 photon cross section in Goppert-Meyer units, which I think are well, they're obviously centimeter square divided by intensity for the units. But oh, by the way, Maria Goppert-Meyer got the Nobel Prize for that, one of the first women to get the Nobel Prize at the time in physics for this. And our goal then is to have a flux of photon that's high enough to make this process not rare. It said differently, we want to have a cross section that's significant enough for us to do imaging. Okay, so when does that happen? That happens when we have a high flux. So when there's a high flux, there is a large irradiance of photons, then we have a good chance of overlap, and therefore the 2 photon process is more likely to occur. So how is this different from 1 photon? So this is all physics, but how does this relate to imaging? So in imaging, typically, we have our objective and we focus the light to our focal spot. But in 1 photon, the light will excite all the molecules inside this cone of light. On the other hand, when we do 2 photon excitation, because the energy, the power of the beam is always the same, but the area over which this energy is spread is getting smaller and smaller and smaller. So the cross section, the fluorescence cross section increases. It reaches a maximum at the focal spot, and then it goes back down. this is a very significant difference from one photon excitation, because we only excite the focal spot, whereas in one photon, we excite everything under the sun. Okay, so for imaging, how is that useful? When we do imaging, with multiphotonic excitation, we shine light, and then we know that only the part at the focal spot will be excited. So contrary to confocal, which I will highlight in a second, we only generate light from the focal spot. Then the light will be emitted, and it may be scattered all over the place by the cells, but it doesn't matter as long as we have an objective that can collect all this light. I know that if there's a green photon that is making it to this area and making it to my objective, I know that I'm only generating photons from the focal spot. Therefore, it's a photon that I want because I know where it's coming from. I alluded to that at the beginning. I said scattering sucks because it prevents us from determining where a photon came from. In this case, because of the physics, we know that the photon is coming from the focal spot, so we want it. So we have in multiphoton microscopy, even if the photons are scattered, they will still be useful. This is really important. This is really the most important part in multiphoton microscopy, because the fraction of light that is scattered but is still detected is very, very high, and the deeper you go in tissue, the more true it becomes. Okay, this is the most important part. So in one photon versus two photon, a reminder, we shine light, we excite everything, but we have a 4F system that will take this point here and will image it onto a pinhole. So at the detection, we remove the photons we don't want. For two photon excitation, that's not the same. We shine light and we only generate light from the focal spot. So there is no pinhole here, no pinhole, no pinhole, okay? So this is very important because the detector can be a large detector that will detect all of the light, and this is really helping for deep tissue imaging, and that's why multiphoton excitation is associated with live animal imaging, because of the scattering and how it actually benefits from scattering at the detection. We're still hurt by scattering of the illumination towards the focal spot, but at least at the detection, we're not affected as much, okay? So the comparison of one photon versus two photon, the one photon excites the whole area. Optical sectioning is obtained from discarding the light that we don't want, and the light, any light that is scattered is just hurting us. If light is emitted in a different direction and then coming back in our sample, it's hurting us. And two photon will only excite at the focal spot, which is great because that means we're dumping energy in the tissue in just a small area. Longer wavelengths penetrate deeper. So this is also helping us. So not only the scattering is helping us for the detection, but the scattering is lower at longer wavelengths. So that's good. Okay. So in fact, the scattering coefficient is low when we excite, and it's high when we detect because the fluorescence is at a shorter wavelength. So scattering in this case is still hurting, but at least it's going in the directions that are helping us. Okay, and if you want numbers, I love numbers. You will see that fluorescence, one photon fluorescence cross sections are roughly 10 to the minus 16 per square centimeters. This is the same as the, if you look here, we have a table of fluorophores on our website, but the biologists often prefer to use the absorptivity epsilon, but they're equivalent. They're essentially proportional to each other. So you can find numbers for fluorophores on the website. And I absolutely love this paper by Drobyshev. It's a Nature paper that's not particularly exciting. If you look at the first few pages, it's very generic and boilerplate, but it has 20 pages of supplementary material with cross sections from the cross sections in 1 photon and 2 photon across a range of wavelengths for 300 gazillion fluorescent proteins. So let's take a quick look. Okay, if we look at let's look at GFP here. I drew too large. So on this side, you have the cross section in GM in Goldberg-Meyer units. And on this side here, you have the absorptivity, which is really the cross section in 1 photon. So what do we notice when we look at this? The GFP in 1 photon, okay, the units for the 1 photon graph are right here. And imagine that there's another axis here that's divided by 2 that should be here on this graph. I don't know why it's not there. But the point is you excite at approximately 950 and a bit in, I'm sorry, at 488 in 1 photon. But in 2 photon, it's closer to, it's not quite 488 times 2. Okay, it's a little bit lower, it's blue shifted. So the optimal wavelength in 2 photon is not necessarily twice as large as the 1 photon fluorescence wavelength or excitation wavelength. I'm sorry. So GFP is a very good example because it is shifted by nearly 50 nanometers. So it does make a difference. If you tune your laser to 980, which is 2 times 490, it's not going to be as you would be here. So if you move over to 950 or 920, you'll have something that's much better. Okay, so take a look at that if you need for your own experiments. And as if this was not enough, this is 1 photon and 2 photon, but now people talk about 3 photon excitation. We could talk about that for a long time, but the point is that imagine that many of the benefits of 2 photons are translated to 3 photon, but 3 photon eventually, when you reach very high intensities in 2 photon, you eventually start exciting the first top layers that will emit in your objective, and this reduces the contrast. And this is true when you go to very deep tissue with very high power. In 3 photon, that effect is smaller. So the benefit of 3 photon imaging is that you get less background fluorescence from the top layers. Okay. All right, so this was my my digression towards 1, 2, and 3 photon imaging because they relate to the wavelengths, but they also relate to the imaging technique. So let's get back to contrast. Okay, there are a lot of endogenous contrast mechanisms that we can use. There is endogenous fluorescence, there is absorption, as Michel was discussing. You can do what's called second harmonic imaging or second harmonic generation, where oriented tissue like collagen can emit color that is exactly perfectly half the wavelength of the incident light. It has been used also to measure the electrical potential across membranes by Dombeck and Webb, but this was fairly difficult, so it never really deployed in all labs. And there's something also called third harmonic imaging or CARS imaging that I will discuss in 2 slides, really. But the point is that it's possible to get some contrast from the tissue itself without adding anything. Just to give a tiny little bit, one or two slides about what we do in my lab. We do coherent Raman. What is coherent Raman? Coherent Raman is when we take two lasers, we take a molecule, we drive it so that it starts vibrating, and the vibration is such that it will give the energy of the vibration to another photon. That's my one sentence explanation of cars. How can we see that as an extension of Raman? Raman scattering, you shine light and the photon leaves some energy in the molecule, and the difference in color is an indication of the molecule. Cars, it's different. You use two lasers that are tuned perfectly to drive a certain vibration. So it's more complicated than Raman, but in the end, you get a cross section that is very effective, and it's essentially, imagine it, picture it as a two photon with two different colors. Although that's so not true, but I think it's close enough for most of you. So in the end, we get light that is emitted that will depend on the vibration from that is available in the tissue. In our case, we have used this to image lipids a lot, and that's the only slide from my lab today. But there's a lot of lipids in myelin, and therefore cars imaging has become a little bit synonym of myelin imaging, but we also use it for spectroscopy in many things in my lab. But this is not a talk about neurosurgery, so I will not discuss it. The point is that we get all the advantages of multiphotonic excitation. We can do optical sectioning, we can get nice high resolution images of myelin, and then use this to infer the health of the myelinated axons. All right, that was it for the slide from my group. So another challenge when we do live animal imaging, is movement. Of course, if you're trying to image something that's alive, sometimes it moves. Breathing is going to be a problem, because we are trying to do very high resolution imaging. So any small displacement will make the image blurry if we acquire the images too slowly, or at least it will move the image if we acquire the images quickly. So the first part really is to make sure the images we get are high quality. So you need to acquire images quickly. So we do a lot of video rate microscopy in my lab. Why? Not because, well, partly because we like fast stuff, but mostly because you need fast imaging to do live animal imaging. If you don't do live animal imaging, if you don't do fast imaging, then it will be problematic. So let me give you an example. If you have on the left, you have a retina that's moving, and if you correct for the displacement, then you can take an average of all the images and get a nice image, even if the animal was moving. So you can either compensate. You can, if you can't compensate because the animal is moving up and down, then you can drop the images when they're not in the right plane. Or in the past, we have made a device that was following the movement of the animal, and then could compensate. So if you average images, you gain in signal to noise, because the number of photons you have will increase. If you have 100 images instead of one, you'll have 100 times more photons, and 100 times more photons means a better signal to noise ratio, because the signal to noise ratio is dependent on the square root of the number of photons. So what did I just say? If you have a single image that looks like this, the best thing to do is to average several images to get more photons and therefore less noise. But if you try to average something while it's moving, it's going to look like crap, because it's all going to be blurry, because the photons are not coming from the same spot, and you're just adding everything. So you need to do a compensation of the displacement. I'm sorry. Sorry. Okay, so if you compensate the movement, then you will have an image that has a better signal to noise ratio without the loss of the spatial resolution. As I said, you can also just acquire several images, get rid of those you don't like, and then keep the final image as the average of all the images that you think are close enough to the one you want. So we have done that in the past too. It works quite well. And the final thing that is possible to do is to have a device that follows the animal. This is obviously more complicated, but it performs better. Why? Because even if the animal is moving left, right, up, and down, we can follow the cells we want without having any time gaps in our movies. So we had made a device where we shined a secondary beam in the sample, and then the reflection of this beam would move to the left when the tissue was going down, and would move to the right when the animal was moving up. So we use that as a feedback system to move the objective with the animal, and then get when the movement compensation is on, it appears like the cells are not moving. And then when there's calcium activity, you can measure the activity without time gaps. All right. So, so different strategies, but you need a strategy for movement, or you need to have a very good surgery, which brings me to my next point. How do we access these various organs? We can't image that deep in tissue, okay? Even with, we say 3-photon microscopy will image deeper, but it's not like if it's going to image 3 centimeters. It may image, let's say, large hundreds of microns into tissue, or maybe a millimeter, but it's certainly not going to be 2, 3, 4 millimeters, okay? So we need to have access to the tissue, and one way of doing it is with surgeries. So actually, no, there are 2 ways of doing this. You may want to do longitudinal imaging, where you image the same animal over time, let's say over weeks or over days. So this would be longitudinal imaging. Or you can have terminal imaging, where you do a surgery, you image what you want, and then you sacrifice the animal. So longitudinal imaging is sought after very much these days, because we can follow the specific details of a disease. So these require surgeries, and most of them are done with what are called windows, as Michel was describing earlier. And there are various ways of doing it. We have developed a spinal window, where you perform a surgery, you clamp 2 metal things on the side of the spinal cord, you put a cover slip, and then you can image through that. The animal has this on its back for a few days. You can also do what's called a cranial window. This is very similar. You get, again, you open up the skull, you drill a hole, you put a cover slip, and then you image over time. Those are not as simple as you may think, or maybe you think they're very complicated, and then you're right. It is actually, it does require a very good surgical techniques and very good methods to make sure that you get a window that doesn't get blurry over time. If you damage the tissue too much, there will be scar tissue that will grow, that will, after a few days, will just block everything, and you can't image through. So there are a lot of tricks that are important to do that. And we do have people here who can make these nice windows, but there aren't tons of people who can do that. So windows. You can also use small micro endoscopes. People may know the small Grintech endoscope. It's a tiny tubular lens that can actually that is so small that it can be inserted between the discs of the spinal cord. It can also be inserted inside the brain, but it's still fairly large. So in the brain, it's hard to, in the brain of a mouse, it's hard to send this in the tissue without damaging the blood vessels and killing the mice. But it's feasible, but it's a bit hard. The other option really, not the other option, but the how this is used is really, instead of using your objective that has a very small working distance and really needs to image through the tissue, you take another objective and then you add this, you put it at the tip here, and then you relay your focal point a centimeter further at the other tip of the endoscope. And we have used them in the past, and we've shown actually that in our case, it was totally fine, and we would get images that were very similar to what we were getting with a microscope. So these microendoscopes do work well to extend when you have enough room to go through. And how do they work really? We have discussed that before. We have discussed that on Monday. It's really just a 4F imaging system. The GRIN is a 4F system. It essentially has two lenses that are acting like a relay and taking this point here, imaging it here, imaging it here. So when you put it at the tip of the of your objective, you can, if you're scanning at one tip here, you will scan at this tip here and you'll just relay the information below. Or you can have a regular microscope with a camera that will just image. You have your objective that's imaging here and you relay, you conjugate these two planes and you can get an image deeper. This is the second strategy that I just described, has been commercialized by Doric Lenses here in Quebec City, where they have a very, very tiny microscope that uses this as the objective and can stay on the animal for a long time to allow longitudinal imaging. Okay. Now the another challenge. Oh, I see that people may have written stuff in the chat window. Yeah, okay, a gazillion. Okay, I'm a bit late. Okay, so the I believe fifth challenge is the size of the sample tissue. This is sometimes that I feel or I encounter is often neglected. Live animal imaging means you're going to be going, instead of taking the tiny slice of tissue that you want to image and image just the small cells, the few cells you want to see, you have the whole tissue. That's great. But that sucks because that means you have the whole tissue. You have to find what you're looking for in the tissue and go back to the same area every day and perform your studies in a way that is statistically significant. So this is hard. And not only do you need to get good data, but you need to analyze the data. So I'll discuss these two things. The first one, again, the answer to large volumes of tissue is to have a fast microscope, because with a fast microscope, you can quickly scan around. Instead of having many microscopes will provide images with a frame rate of 4 images per second. This is fairly common, but 4 images per second is fairly slow. You're trying to scan your sample. You're trying to look, you're looking for something specific, labeled cells, etc. So having something that's fast is really important. At the same time, it's not enough to have something that's fast, because I could give you something that's very fast, but only images a tiny little area, let's say 10 microns by 10 microns. So then it's not looking for a needle in a haystack. It's looking for something in a haystack with a needle. You know, that's going to take forever. So you want to have a large field of view and a good speed. So is it possible to have a very large field of view with a good resolution, with a good speed? Yes, it is possible. If you remember, we have discussed just a few minutes ago that, and Monday, that the resolution in a microscope is given by the numerical aperture. So you want to have as high a numerical as possible to get a good resolution. But we want to have as large a field of view as possible, because we want to see a lot of tissue. Those things are not mutually exclusive. Actually, they're essentially unrelated. The resolution you can get is given by the NA, and the field of view is given by the magnification, and you can have a low magnification with a high NA. It's totally possible. Okay, and the other thing that I think is critical, and I find is sometimes neglected, real-time feedback. If you're in the lab and you're trying to image your tissue, you want to know immediately. So if you can have tools that will look at the calcium intensity, or will look at the number of cells you have per area, etc., getting real-time feedback is very critical. And software we have made in my group and the strategies we've pursued in the group focus on getting real-time feedback as much as possible. Okay, yes, another challenge you may get if you do live animal imaging, you'll acquire videos, you'll do a lot of imaging over an extended period of time. Then you'll have tons of data, and then by magic, you'll just wave your hand like that, and all the analysis will be done. No, of course not. So getting a good putting in place good strategies to process the data is not information. Data, you can have gigabytes of data and 10 bits of information. So processing the data, this is data, this is data, but here this is information, because we have analyzed all the little myelinated cells, and we have found, after having found them, we can look at the thickness, etc. It could be calcium activity, it could be other things. The point is going from data to information requires typically software. So software has many faces. Some people use only MATLAB. Some people use the software provided by the manufacturers. Some people program their own things in Python. It's fairly trendy to do Python these days, which is better than MATLAB in many respects, but MATLAB is also much faster for other things. So you pick your tool of choice. But the point is that many of the things we want to do when we do live animal imaging and live imaging analysis can be done in all of these different software. So for instance, there are things that are ready in Fiji. Fiji is a version of NIH Image, which is the original package. Fiji is really just Image J. That's why it's called Fiji. You can get code that will compensate, that will align stack of images fairly quickly and fairly easily. So you should use them when you can. MATLAB code for registering or aligning images is also readily available and not difficult to use. So you should do that. But sometimes we have to develop our own solutions. So for instance, in our case, we have developed strategies to segment myelin images. All I want to say is that if you're trying to get your own solution for doing an image analysis, it's totally possible. Okay, it is possible, and there are a lot of tools. But if you can use those that exist in package that can be readily available packages, then you should, because on the web you will find tons of examples for automatic segmentation, for instance, to find cells. But these are just trivial examples with grains of rice that are not touching or cells that are not touching. This is garbage. Real life is not like that. So if you can use solutions that already exist, that's good. If not, it is an area of research. We are developing many things like that in my group, but it's not necessarily simple, because the simple strategies where you just want to segment by thresholding and then that doesn't work. Okay, there are fancy techniques, and they are more complicated, like watershed snake, others. These are all things that are known to people in the field, and you may not be an expert, but very quickly when you get into automatic image analysis, then you need to go deeper into these techniques to make useful software that is not applicable just to your situation, but to more situations. So yeah, I could discuss how to make good software, but that's that's for a different time. By the way, I will be giving, with someone from my group, I will be giving a Python for beginners workshop in a few weeks. Ask the Servolet for more information for the dates and the Zoom link. All right. And if you are interested in getting numbers, getting stuff that can help you make a decision, you can take a look at our GitHub, the DCC Lab GitHub. We have, as I mentioned earlier this week, we have a ray tracing package that can model your optical systems. And we also have this Monte Carlo code that will give you numbers for how many photons are absorbed, how many photons are scattered, what's the amount of power that's coming out, et cetera, et cetera. And we have another package we're working on that's not quite released, but it provides you the basic tools to do basic image analysis fairly easily in Python. So those are all the challenges we want to address in live animal imaging. And I have a single application example. I removed 40 slides that I would have otherwise presented, but I will just discuss this the idea that in some diseases, like multiple sclerosis is a disease that we have looked at because we had this great technique that could image myelin. So we thought maybe we can start using this technique to follow the myelin degeneration in the tissue. And as soon as we started imaging the myelin, we also noticed that imaging other things, including the cellular infiltration in the tissue, would be helpful. So we have put together a few techniques that allowed us to look at the timeline of demyelination in tissue. So let me just talk about that for a few slides, really. when you have a disease like multiple sclerosis or a neuroinflammatory disease, you typically have cells, immune cells, that are in the blood vessels that will expand outside and will stick to the blood vessel walls and eventually go through and enter the tissue. So this is given by when this happens, when this inflammatory situation occurs, the blood vessels will become permeable. So it's possible to measure this permeability to get an idea of when the inflammation is being initiated and how this will affect eventually the degeneration of the tissue. So let me show you, if you look at the top here, what you see, we inject dyes in the blood vessels. So in this case, we have inject a green dye and a red dye. So I will not really get in the details, but there's a Texas red. The red dye is called Texas red with a dextran, but it's Texas red. And there's another one called fluorescein or FITSI that is green. So the green dye is a very small molecule. The red dye is a very big molecule. So if you inject them in the tissue, in the bloodstream, then after some time you'll start having sometimes the red dye will leak out, and sometimes the green dye will leak out, and sometimes they will both leak out, but they don't leak out from the same areas, and they don't leak out with the same dynamic. So we have used this to look at the permeability. So we suspect that the two dyes, because they're not the same, so as I said, the Texas red dye is a very big dye and the fluorescent dye is a very small dye and it's charged. So we expect that the mechanisms are different. So if we use an animal model of multiple sclerosis, I'm totally skipping the details here, but with live animal imaging and this technique, which is non-invasive, we just we inject the fluorescent dye in the bloodstream, which is just a simple injection. And then we look at this, we make many of these images everywhere in the spinal cord or in the brain. And then we collect the percent permeability with a very fancy formula we developed. And after that, you can get this graph that doesn't look like much, but took months and months and months of work, where you have the amount of permeability over time. So this is not possible without live animal imaging. You cannot, it would be extremely difficult to obtain this type of graph because the permeability, if you measure the permeability in an animal and then you sacrifice the animal, you won't know if the animal got sick the next day because you killed the animal. If you do live animal imaging and longitudinal imaging, then it is possible to image the same animal every day. And then eventually you will get, let's say on Wednesday, you'll see that the animal, because the animal, you put the animal back in its cage and you monitor if it's getting sick. If it's getting sick on Wednesday, you can actually go back to your Monday images and call these images the permeability 2 days before the animal got sick. Okay, so all of this is possible thanks to live animal imaging with good sampling strategies and with good automatic analysis techniques. And so we have used this we have done this for permeability. We have also done this to look at immune cells, how they eventually leak out of the blood vessels. The point is that that's what I'm insisting on here. Okay, look, if you look at this graph, this is an important graph because it's really a graph that can only be done with live animal longitudinal imaging, because we have images, we have information before the animal got sick and after the animal got sick. So I just showed 2 slides really that with interesting results. This is a combination of the right dyes, the right strategies, the right objective with a nice, good field of view and good NA, good analytical techniques, all of those together to make these graphs possible. So I was fast. So it's been an hour already, and I have a few things to say after that. But I will take I will conclude here the first part of the talk. So I only have a few slides after that. Live animal imaging requires adaptive tools that tackle the important problems of tissue imaging. I've discussed scattering, absorption, contrast, the size of tissue, the movement, the access to organs with surgery, and the analysis of data. All of those are important, and you need to have a good grasp of all these aspects if you want to get good live animal data. Developing a good surgery protocol takes time. It actually takes a very long time. Okay, developing a good imaging protocol in live animal is the part that is forgotten by everyone when they start live animal imaging. Because if you do Petri dish imaging, you take your cells, you put them in the dish, and you image them. For live animal imaging, it is more complicated. You need to have a good surgery every day. You need to have, let's say in the example I gave where we inject the dye in the bloodstream, injecting a dye in the bloodstream is done via a tail vein injection. It's not easy. Not everyone can do that. So all of those need to be done together in order to get good live animal data. All right. Do we have any questions by any chance? Was I on mute for the last hour? Okay. Anyone? No? That's okay. You can wait after. You can. Yeah.
I had a question, but it was a question for way back in your presentation.
Yeah, that's okay.
When you speak about NA and magnification, like if you compare a 20x, let's say, with a NA of 1.1 versus a 100x with a NA of 0.9.
Yep.
Will you still get a better resolution with the 20x? Let's say you have a CCD that has enough size to, or your resolution will be better with the 100x. Sorry for the question.
Yeah. It is. I was trying not to talk about that, but what I discussed is the best resolution you can get is given by the NA. But then if you are using a camera, you need to make sure that all the pixels are roughly the size or smaller than this spot at your the spot you're creating with the objectives. What am I trying to say? With the higher the NA, the higher the best resolution you get with the best NA. But if you don't choose your camera properly and you only have a few pixels, let's say 100 pixels instead of 1,000 pixels, then the pixels will be the limitation. So when you are making a system that has a very high NA, you need to make sure that the pixels you have per distance is sufficient to cover the resolution given by your NA. Is that clear, or that's too?
I think that's clear, but like if you have a 20x with one NA and you have and you have a 100x with 0.9 NA, and you use the same camera
Mhm.
on the one before, like you use a very, let's say the camera is not the problem, it has enough pixels. Will you be able to numerically zoom on the 20x and still have better resolution than on the 100x? That was my question, actually.
Yes.
Yes.
The
Okay.
answer to that is yes. It may be worth a longer discussion, but the thing you will want to look at is the parameter that's really critical is the field number of your objective. This is something I very quickly discussed on Monday, but the field number is essentially the size of the CCD that you need to use that works with this objective. So I very much suspect that the 20x with a very high NA and the 100x with a different NA probably don't have the same field number, but I'm just guessing. So yeah. So that's what will determine, and the field number they quote is the optimal field number. So if you use them with I guess what I'm trying to say is that the manufacturers will give you a camera that will work with all the objectives. If you're making your own, you do have to worry about the pixel size of your camera, how it is imaged at your sample. So you calculate your pixel size, you have the magnification, let's say it's 100. Therefore, your pixel, let's say if it's 5 microns, the pixel at the objective will be 0.05 microns. So you have to take that into consideration.
So that way you'll get the most out of your optical system, but you would still get a better resolution with the 1 NA instead of the 0.9.
Yep.
Okay,
Yep.
Thank you. That answers it.
Good. All right. So, whoops. All right. Okay, my green screen is moving. Okay, so I really like this part. I have a lot of common lab problems that I want to discuss with you. So maybe some of them you may have seen before. So the one is, for instance, the photomultiplier tube, the PMT is fine. It's fine. I checked it, boss. It's fine. Well, is it? Is it? So there is an ideal voltage for a PMT. Let me repeat this. There is an optimal voltage for a PMT. I help other people in different labs, and the first thing I look at is the voltage of the PMT. A PMT will amplify all the photons and give you a current. If you don't amplify well with the right voltage, the signal will be too low and you will not see the photons. So you want to have a voltage that's high enough to be above, you don't want to miss photons. But at the same time, if it's too high, then you'll start having photons that get spuriously emitted and amplified at different stages on the diodes. So you need to know the optimal voltage. I know that for the multi-alkali of PMTs that we use, that voltage is just under 1,000 volts. It's 900 volts. So typically you want to have the voltage at 90 to 95% of the maximum value. Okay. Very often, people in the lab will, if the signal is too high on the screen, they will reduce the PMT voltage because that's the easy knob that we all have. It's right there. It's on the software. You just lower the voltage. But that's not optimal. What you really want to do when the signal is too high is to reduce the laser power. because you will damage the tissue less, and the PMT will see all the photons that are available. So voltage of PMT, I will say 90 to 95% of what's available, and laser power as low as possible. So the PMT is fine, yeah, but are you using it properly? Okay, now, whoops, now, no, not this. Okay, now, boss, I found this avalanche photodiode. Oh, it's much better than the PMT. The quantum efficiency is 90%. This is much better than the PMT. The PMT has 15% quantum efficiency. So I think we should use APDs. Well, should we? There are three things that matter when we make measurements. How big, how large the signal is in volts per photon. So the signal, the number, I wrote per milliwatt per photon. That's the, yeah. So how high the voltage is for a given number of photons. And the quantum efficiency is something else. The quantum efficiency is how many, what's the probability of a photon hitting my detector and generating something. So in an avalanche photodiode, 90% of the photons will hit the detector and will generate one electron. So they are technically detected. The quantum efficiency is high. In a PMT, it's much lower. The multi-alkali PMT has a quantum efficiency of 15%, which is very low. So the quantum efficiency of the APD is higher. So one photon generates an electron more often on an APD than a PMT, but this is not the final signal. The electronic gain you have in a PMT is 1 million, whereas in an avalanche photodiode, it's only 50, 100, 200. 200 is actually very high. So the signal in volts is actually lower in an APD than it is in a PMT, and that makes it more difficult to detect. So the electronic noise in the signal, the electronic noise in the detector in an APD is such that many of the photons we don't see because the signal we get in millivolts is lower than the noise, whereas in a PMT, the signal is above the noise and we can see very low optical powers. Okay. All right. Yeah, okay. So a PMT is more sensitive than a PMT is more sensitive than an APD, and that's why we use them. Okay. All right, boss. There's way too much autofluorescence in the sample. It's not working. It's not going to work. The grant you wrote, it's not good. Well, really. If there is autofluorescence, well, maybe the fluorescence that you get from the endogenous molecules in your sample, maybe it's at a different wavelength. So if it is at a different wavelength, then maybe we can remove it with filters. Maybe we can use a different excitation wavelength. This is actually a surprisingly useful advice. Maybe the optimal wavelength to get the best, the most intense signal is, say, 900 with a Thai sapphire laser for some experiment you're trying to do with a GFP. But maybe when you excite at 900, you excite a lot of other molecules that are already in the tissue, and you could move to a different wavelength. Maybe you'll reduce your signal, but maybe you'll reduce the background even more. So choosing the right excitation wavelength can have a very important effect. This is something you do at the beginning when you start with your experiments. And the final autofluorescence problem is really the background light coming from the room. Monitors the small flashlight you use to look to read your lab book, the room lights, the warning signs. I had that in one of my projects. There was a red light, a warning sign that was blinking inside the room, and we could see the blinking in the videos. So there are a lot of things that can happen. So background light, those detectors are very sensitive. So using a good isolation is very important, and the light can come from various. You have to inspect your setup to make sure you see where the light is coming from if it is spurious light. The spurious light that doesn't have to be there, you remove if you get a chance. Okay. Then this this red fluorescent protein, boss, it doesn't work well. I tried it and it doesn't work well. Well, really, just because your microscope says red filter, that doesn't mean it's good for Texas red. Okay, what does it mean, red filter, in your microscope? It means whatever it meant to the person who labeled it red. Okay, which may not be what you mean. So always checking the fluorescence cubes or the dichroics is very important. You have to check the exact specifications of your dichroic that are reflecting the light, the emission and excitation spectra. These are the filters. So we often talk about the dichroic. That's the most critical, I would say. But I've seen there's a little bit of, not a little bit, there is redundance in these filters. There's always a filter that will clean up the illumination to make sure it's in the range we want. And there's another one called the emission filter that will always reduce or remove any light that should not be there. Even if the dichroic is really good, you still need this filter, the filter to block to clean up the illumination and to clean up the emission. So these filters sometimes are too restrictive. So we talk about the dichroic, but are we really talking about the other filters? And as I said last time, the light source you have also could get blocked by the emission, by the illumination filter. Okay, so always something to look at. What's in the cube? Typically, when I help other students, when I go in the lab in other groups, and they have issues with too weak a signal, the first thing I'm going to look at is the cube. You know, what cube are you using? I don't know, the red one. Okay, well, that's not good. Finally, I have two things to discuss. Yes, this objective is not that good, boss. You paid $20,000 for that, but it's not that good. I tried it, it's not that good. Well, we have to use the proper cover slips. The cover slips, when we need a cover slip, we want to use a cover slip. You must fill the back aperture, which is probably less common. It's not a problem with people using commercial microscopes because they tend to be well designed, although that's not always true. But if you make your own, you have to fill the back aperture. So to make sure you're using the full NA of the objective, and everything that I just said with using the cover slip, making sure you fill the back aperture. When you do single photon imaging, you can excite everywhere, and then the pinhole will just remove what you don't want. So the signal may be low, but you will still have signal from your focal spot. When you do multi-photon excitation, if the objective is not used properly, for instance, if you put a cover slip when you shouldn't have, then you will have a poor focal spot and therefore poor generation. So low signal and problems. So in multi-photon excitation, 2-photon, even 3-photon, second harmonic, all these multi-photon strategies, they are much more sensitive to aberrations and, let's say, poor use of the objectives. So, so that's something you want to make sure you do properly. And the final slide that I have, this protein, boss, it sucks. It photo bleaches too quickly. We're not going to be able to do the experiments. I'm sorry. Oh, really? Well, when your dye photo bleaches, that means that the fluorescence goes down over time. So you're illuminating, you get some light, and then after a minute it goes down, and another minute it goes down. So it's because the laser power is too high. Well, typically the laser power is too high, probably because your PMT voltage is too low and you crank up the laser power to compensate without knowing that this was not equivalent. So it photo bleaches, you have to lower the laser power. That probably means increasing the PMT voltage if you have lowered it before by mistake, okay? Maybe you don't have the right dichroic. Maybe your dichroic sucks and you're filtering some of the emission and therefore you're increasing the laser power to compensate. Maybe you're cutting off too much of the fluorescent spectrum. Maybe you use the cover slip or even maybe the wrong cover slip with your objective, and all of that is leading to lower emission, and you think that you have to crank the laser power and you photo bleach the dye. So the real problem is not photo bleaching. It's just that your laser power is too high because your collection efficiency and excitation efficiency is too low. All right, so this was it. Okay, so stop sharing right here. Here I am. Okay, so 12:25. How about that?
Excellent
So
job.
do we have questions? Do we have questions? Well, you may have questions later. I'm sure that a lot of people heard things that they already knew, so I'm glad you feel super intelligent now. I'm around. Feel free to ask questions. If you don't have any questions now, well, you may have questions later. Okay, so we're done here.
Thank you very much, Danielle, for once again an excellent talk.
No problemo.